as said, my name is Teresa Cunha, and I am a PhD student at the Technical University of Denmark and also the Danish Research Center for MR. So we're here based in Copenhagen. And today I will focus on how to do concurrent tES-fMRI, but from a technical point of view. So I will tell you about safety and image quality issues. And on this image quality topic, I will present you some of the work I have been doing together with colleagues from the Free University of Berlin in Germany, since we are all part of this large research consortium called MemoSlab. I have a background in engineering, and I think that will be fairly clear from this talk, but I will try to explain the basic concepts so that everyone can follow along. So this initial part of the talk was heavily inspired by a consensus paper on best practices for both performing and reporting tES-fMRI studies. I collected a pair of quotes that I think summarize quite well the need for having this talk. And so we start by claiming that we can combine tES with fMRI to map brain activity during neuromodulation, and by that support causal studies of brain function and tES effects. The problem is it is not as straightforward as it sounds, because combining these two techniques turns out to be quite challenging from a technical point of view. Furthermore, and because of that, the results of these kind of studies become highly dependent on the methodological aspects of the study. And so it is important to properly report them to ensure that the results are reproducible and that we can interpret them properly. So this paper collected inputs from over 50 experts in the field to make a list of recommendations for ensuring safety and data quality, but also for giving guidelines on how to report these methodological details in a more systematic way. If you do follow the recommendations present in this paper, you are on a good track to avoid safety hazards, like, for example, RF burns under the electrodes. Yeah, and also image artifacts like the ones you see here, and we will see in more detail later on. I do have to make a note here that so far RF burning artifacts under the electrodes have never been reported in tES-fMRI studies. Very likely because the community is well aware of the hazard and only uses suitable setups. So this was just an example image from a burn under an electrocardiogram electrode, actually. But even if you do so, you follow all these recommendations, you don't have safety issues, and your images look nice. You may still have not so obvious artifacts in your data that can cause false positives in your fMRI analysis. So my talk will be divided into two main parts. I will start by showing you what a typical tES-fMRI setup looks like. And then I will convince you that your protocol should also include some scans for visualizing the electrodes and cables, because they can give you some artifacts, as we will see. Then I will explain you what makes electrodes and their leads MR conditional or MR compatible, and why this is needed. And on the second half of the talk, I will focus on the main types of current-induced artifacts on EPI time series. I will list three here, although we will see in more detail what each of them mean. And then I'll wrap up this part of the talk by proposing an adapted acquisition and processing pipeline to mitigate these artifacts. I will finish the presentation with a pair of take-home message slides and just try to initiate some discussion as well. So, for doing tES-fMRI, the basic technical requirements are an MR scanner, obviously, and tES equipment, which should be MR conditional. Then, if you want to evaluate the online effects of your stimulation, you also need to make sure that you know exactly where the stimulation occurred within your fMRI series. So we can use a trigger module so that the scanner sends a trigger to initiate stimulation. And you will also have to then connect the electrodes, the scalp electrodes, which are inside the scanner together with the patient, with the MR stimulator, electric stimulator, I'm sorry. Despite being MR compatible, has to stay outside the scanner room. And as many of you probably know, the MR room works as a Faraday cage, keeping radio frequency signals coming from the outside, preventing them from coming inside, because what we're trying to measure in MRI is also radio frequency signals, which are extremely weak. So having something coming from the outside would disturb our measurements. And so just drilling a hole through the wall to make the cables go through would ruin this cage, and you could have artifacts like these vertical stripes that I show here. Let me get my pointer here. So one way to address this is to filter your signal. And there are two main approaches. The first one is with a pair of filters, one outside the scanner room and then another one inside, which has to be MR compatible as well. And then those filter boxes would be connected with a wire that would go through a waveguide, which looks something like this. This is not very efficient against common mode noise. It can be improved if you connect one of the filter boxes to the waveguide, for example, by clamping it, so that they share the same ground. But the preferred solution would be to use an RF penetration panel that looks like something like this, which has already inbuilt filters. Lastly, even when you do this, you should run frequently some RF noise measurements or spectra just to make sure that everything is working as intended. Finally, you will have to bring your electrode leads and wires out of the scanner bore, and while doing that you should try to avoid creating loops of highly conductive material, because there is the risk for electromagnetic induction in these loops due to the very fast oscillating magnetic fields sent out during excitation. The next topic in our agenda is about electrode visualization and their artifacts. And it turns out that visualizing electrodes is actually quite tricky with MRI, because first they don't produce a strong signal, and then that weak signal decays extremely fast. So we need to use a type of sequences called ultra-short echo time sequences. And a very popular one is called Petra, which is the one that produced the images I showed here. And the reason why you would like to see where your electrodes are with respect to your fMRI data is because the material from which they are made of can cause artifacts in the data. And that is because that is through the induction of inhomogeneities of the static magnetic field B0. For example, what you see here, when you compare the situation with electrodes and without electrodes. And these inhomogeneities will cause susceptibility artifacts in your EPI images. And it will also induce inhomogeneities of the B1 field, the field involved in the excitation, so that you will not have exactly the same flip angle throughout the entire field of view. So here I just show some images from a different paper where you can clearly see signal dropouts and some distortions under the electrodes. Same thing here. And fortunately, these distortions are typically restricted to the scalp and skull regions, so they usually do not reach the brain. But even so, it is good practice to have a scan while you can, a scan where the electrodes are visible and where you can overlay your fMRI data on, so that you can then know where to look for these artifacts and make sure that your image is actually doing okay. And if you have, this is the last advice, if you have both sham and active conditions in separate scans, then you should try your best to make sure that the electrodes were exactly in the same position, because if there is some artifact, then it would be common to both sessions. So we've just seen some image quality concerns associated with the electrodes, and now we'll see that there are also some safety concerns, but they can be avoided to a great extent by using MR conditional equipment. So when we have a conductive material in a time-varying magnetic field, you will see the induction of some current loops that we called eddy currents. And in MRI, they are caused both by the fast oscillating transmitted field during excitation, but also to the slower but also time-varying imaging gradients. So the way to address this is to use, I also forgot to mention, this is mainly a concern under the electrodes, on the electrodes, I mean. And it can cause heating and also give you artifacts. And so a way to avoid this is by using lower conductivity, smaller and thinner electrodes, and also to avoid direct contact between the electrode and the skin by applying a general layer of conductive paste in between the two. When it comes to the electrode leads, the main concern is the antenna effect, which I'll try to explain. So when you have elongated materials, highly conductive materials, they will pick up the electrical part of the transmitted RF field. They will redistribute this energy and typically concentrate it at the tips, which we show here. And so when you have conductive wires, for example, made out of copper or carbon, you would have to add some safety resistors to decrease their conductivity, keeping in mind that there might be strong electric fields close to these resistors, so they should not be directly contact the skin either. To further minimize the amount of conductive material that you have close to the subject, you typically bring the electrodes out of the coil from the top in such a configuration, which improves safety on the one hand, but has a caveat. So we know from the Biot-Savart law that a current flow is capable of inducing magnetic fields. And so this kind of cable configuration induces magnetic fields that are not at all negligible, and that can give you artifacts in your data. So to address this, a former PhD student in our group developed these low conductivity silicon rubber electrodes, which were shown to pretty much almost entirely avoid the risk for the antenna effect. And so we no longer have the need to avoid contact with the patient. So we can bring the cables straight along Z in order to avoid these undesired magnetic fields. Of course, depending on where you want to put the electrode, you will not always be able to avoid some bending, and so some of these undesired fields, and they can induce distortions, which is what we're about to see. So this brings us to the second main part of this talk, which is about the artifacts caused by these current-induced magnetic fields. And as before, I start with a very important paper on this topic, already from 2012. But to the best of my knowledge, it was the first and only paper where they did a proper investigation of the impact on the Bolt signal of these current-induced magnetic fields induced during transcranial electric stimulation. So what they did was they applied tES-fMRI in 2 postmortem subjects, and they observed that the size of its effects on the EPI series was of the same order of magnitude of the Bolt effect in a normal finger tapping experiment. To better understand where these artifacts would come from, they simulated current flow inside the brain and computed the induced magnetic fields and observed overall good correlation between the magnetic fields and the activations observed. However, they could not explain the flip of sign in the activations here in the ventricles, and they also did not try to give a formal description of where these artifacts would come from a MR physics point of view. So our colleagues in Berlin, who also do tES-fMRI, got very intrigued by these findings and reached out to us in Copenhagen to help them investigate these in more detail. So together we started with a tES-fMRI experiment that looked like this, in vivo, but in resting state, to make sure that there could not be any bold activations, and all the effects we observed in the data would be artifacts coming from the current-induced magnetic fields or current flow inside the brain. So we ran a very simple GLM analysis using this, the current injection scheme as regressor, and to our surprise, we got these beautiful activation patterns. So we asked the question if these effects could really be induced by the current-induced magnetic fields, which are extremely weak. If you see here, we did not go over 2 milliamperes in the simulation. So weak currents generate weak magnetic fields, but could they be strong enough to induce considerable effects? And if so, since we're dealing with EPI images, our first intuition was that these effects were caused by geometric distortions, but could there be something else? And finally, what was the main source of these problematic magnetic fields? The currents flowing inside the brain, or the ones flowing in the cables, wires? So before I share your findings with you, I would like to spend some time explaining for some and refreshing from others how spatial encoding works in MRI and how flaws in this process can cause the geometric distortions that I have talked about. In a conventional MRI experiment, you have the spins rotating, or as we say, precessing around the main field B0, according to this famous equation. This is the Larmor equation, stating that the speed at which the spins precess is proportional to the magnetic field strength. Now, let's suppose that you have a group of spins at different locations. And if we had the same magnetic field everywhere, they would be all precessing at the same speed. And we would have no way of finding their location. So we can do something smart and vary the magnetic field in a controlled way by applying a linear field gradient, so that some spins will precess slightly slower in here, some others will precess slightly faster. And by these we create a lookup table, or a mapping between precession frequency and position. And that's how we do what we call spatial encoding in MRI. This is a very clever idea, but it's unfortunately not flawless and can become quite inaccurate in some situations. For example, when we have inhomogeneities in the static B field B0. For example, in regions of air tissue interface, so you typically see signal dropouts and distortions in the frontal areas close to the frontal sinuses in EPI images. And unfortunately, EPI is also the standard sequence for fMRI, so it's important that we understand where these artifacts come from. So if we recall the situation from the previous slide where we strategically imposed a linear field gradient to be able to locate our spins, what would happen if our background field, B0, was not perfectly homogeneous to start with? What if it would look like something like this? So when we would apply our linear gradient, we would no longer have a linear mapping between precession, frequency, and position. And if we take a closer look at these three spins, the true magnetic field that they see is actually the red curve, so they would see the same field, therefore they would have precessed at the same frequency, and after a while, when we take a picture of the spins at a given time point, they would all be pointing in the same direction, so they would have the same phase, as we say in MRI. And the problem here is the scanner doesn't know about this. The scanner thinks that we have a straight black line, and when it tries to assign positions based on this, it will make mistakes. So, as I show here, I can go back. It will try to use the lookup table created, and it will assign positions wrongly. And this is what causes geometric distortions in EPI images. So we have seen that, I am telling you that these kind of fill gradients due in air tissue interfaces are quite strong and are capable of causing visible distortions. But are there other sources of field inhomogeneities we should be worried about? What about the magnetic fields caused by the transcranial electric stimulation currents? They are very weak, but maybe they can induce artifacts that are comparable to the bold effects. So to investigate this further, we started by using a software developed in our group called SimNIPS, where we simulated the current flow inside the brain, used these, induced by this montage, and calculated the induced magnetic fields. And as expected, they turned out to be quite weak. So we are in 2.2 nanotesla, which is 10 to 10,000 times smaller than the field inhomogeneities that we expect in the frontal sinuses, for example. And because we know the theory of EPI image generation, we can predict which the distortions that they would induce. And they have two components, so they induce shifts in voxels, which is what I just explained you. But they also induce some stretching or compressing, which will cause intensity changes in the image. So when we combine these two types of distortions, we can have a combined intensity change map, which we see here that it was very small, so 0.02% change intensity. But this was due to the currents flowing inside the brain. What about the currents flowing in the cables? The difference to this situation is that here the currents are extremely concentrated, as opposed to when they are just allowed to spread inside the brain. And indeed, they will induce a lot stronger current-induced magnetic fields. Here we have a factor of 50 from the first to the second row. And when we look at the combined intensity changes, we see that they reach around 1% of intensity change. And this is comparable to bold signal changes. Of course, these were just simulations, so we tried to verify these in vivo. And we tried to, just to make sure that we would have no currents flowing inside the brain, and that just currents in a cable would be enough to cause such artifacts. We used what we call a current loop setup. And now if you paid attention to one of my slides in the beginning, you would probably think that it's not a good idea to use a loop inside the scanner. That would be true, but the issue comes when you have highly conductive materials. And we used the electrodes developed by our previous PhD student with very low conductivities, such that this was not a hazard anymore. So we managed to create magnetic fields comparable to the ones that we had in the previous experiment. And when we do also a simple GLM analysis, we see that there are activations where we expected them, so close to the cables, where the geometric distortions occur. But we also saw activations that we could not really understand. So there were remote effects, especially in the brain interface between brain and background, and also between CSF and brain tissue. So this told us that there was something else besides these local distortions. So we found out that the dynamic updates of the scanner center frequency throughout the API time series were also sensitive to these current-induced magnetic fields. And this probably doesn't tell you much, so I will try my best again to explain in the next slide what we mean by center frequency and why it is important that the scanner is able to update that throughout times. So let's assume now for simplicity that we have no imaging gradients, so we have the same field B0 everywhere, and the spins are precessing proportional to it. When we take a picture of the spins at a given time point, they will be pointing in a certain direction, which we call phase, as you've seen. But throughout the course of an API series, because of heating from the fast switching gradients, there will be a drift on this background field. So it is a slow drift, although I've just shown a fast varying curve, but it is a slow drift over time. And what will happen is the spins will precess either slightly slower or slightly faster. And if we take a picture at the same time after excitation, but at a much later point of the PI series, they will be pointing at a slightly different angle. They will have a slightly different phase. And the important thing here, compared to the previous case, is that all the spins will be affected equally by this. And the net effect of that, when we try to use the lookup table that we created, we will also see a drift on that. And when we try to make the assignments, all the spins will be shifted by the same amount. And these will give global shifts of the image. And you would have something like this, which I believe was common in the early days of fMRI, that you would see your image shifting throughout time. So it's a great thing that nowadays the scanners have the capability of updating the center frequency over time to avoid this. The problem is when it gets fooled by the current-induced magnetic fields and assumes that they are part of this low frequency drift, low field drift over time. And we'll see how. So to verify that, we did another experiment, this time using a phantom, where we tried to create very different magnetic fields in the slice direction. So we had a top slice with strong magnetic fields and a bottom one with almost no magnetic fields. And here I show, we performed two experiments, a standard one where we just allow the scanner to freely update the center frequency, and we also ran another one where we did not allow it to do so. So here the center frequency was kept constant. Here we have a proxy of the center frequency update. And we see that when the scanner was not allowed to update it, there is a drift over time in both slices, which is largely resolved when we do allow it to do its job and update it. The problem is, and I will detrend this series to highlight it better, is that here, when the frequency update was on, we are looking at the bottom slice, and there are almost no magnetic fields there. So why would there be the current injection scheme clearly represented in here? Well, this is because when the scanner is trying to estimate, to have a rough estimate of what the center frequency is at each time point, it will do an average over the imaging volume, such that it will end up not really in a situation where it cannot really correct it fully where it's needed, and ends up inducing it also somewhere else, because there can only be one central frequency. And we see that that is not the case when we did not allow it to update. When we look at the activations that this causes, we see that when the frequency update was off, we don't have major activations in the bottom slice, as expected, because the fields were extremely weak. But we do have activations at the edges of the phantom and in between the different compartments, which are compatible with these global shifts caused by a shift in the center frequency. So this adds to the list of artifacts that we understood. If you recall, we also had the local geometric distortions in regions of very strong magnetic field and field gradients. But even when we had the frequency update turned off, there was still some remote effects that we did not fully understand in regions where the magnetic field was also weak. So this takes us finally to the third and last source of artifacts that we managed to identify, and which are caused by simply applying standard rigid motion correction pipelines. Here we have an experiment pretty similar to the one we showed before. And here you have the activation maps that we obtain when we don't perform any kind of motion correction. Now, when we apply a standard motion correction pipeline, in this case was through using SPM, we will have the activation pattern will change drastically, also in a way that we don't really expect. We don't really know what will come out of that. And this is because when we do a rigid motion correction, so we only have 6 degrees of freedom, either we translate or rotate, we cannot correct for local geometric distortions, because those are highly nonlinear. So what the algorithm will do is we'll try to rotate and shift the image around to best fit the two images, but it will not be very successful. So in some places, yes, it will manage to correct somehow, but in some others, it will induce or invert the direction of signal changes. And this creates artifacts that we cannot explain, are just unexpected. If we look at the motion parameters that come out of this algorithm, we see that the current injection scheme is very well represented here. So we can do a trick, and before applying the correction, we can regress out the current injection scheme from them, and we see that we recover the initial activation patterns, so the ones we understood. This is just an example of how that performed also in the human. So a standard motion correction pipeline would induce activations in the front, where we had the cable in the back, so we would not expect such strong activations in the frontal part. And when we regress the current out from the motion parameters, these activations are gone. So we've identified the main sources of artifacts, and now we try to develop an acquisition pipeline that is robust to them. So we would have to start with scanning without online frequency update, but we would still have to correct that offline, otherwise we would have these drifts in the image. So we would have to extract the raw data from the scanner, and that's the main bottleneck for not really being feasible to do this in practice. We'd have to extract the raw data and do this correction offline by knowing what the current injection scheme was, so that we can regress that out. Then we would have to do the adapted motion correction pipeline that I just showed you. And because we have a scan where we see not only the electrodes, but also the electrode leads, we could track their path and apply the Biot-Savart law to predict the induced magnetic fields, which we would then correct by knowing the theory of EPI geometric distortions. So just to show the results of this pipeline, here in the first column you have a standard pipeline, normal acquisition, and then we have the proposed one. We see that the activations are strongly mitigated. This is not perfect. The same thing when we had current injection. So the main take-home message is we can try to mitigate it on how we acquire, but the best way is to try to avoid them from start by design. So I'm finally reaching the end of my presentation. I just have a couple of take-home message slides, if you are still able to follow.
I wonder if we can take that for the discussion section.
Yep.
We're sort of running up to the hour, so I wonder if we could maybe have a discussion for the last 5, and we could perhaps go through some of the take-home messages there.
Yep.
Would that be okay?
Yes.
Great. Okay. Can I? Well, please thank Teresa.